Cleavage. Shortly after fertilization, the zygote undergoes cleavage, a series of rapid mitotic division with no period of growth during each cycle. There are two reasons why cleavage is important. 1. Generation of large number of cells that can undergo differentiation and gastrulation to form organ. And 2. Increase in cytoplasm versus nucleus ratio. Cleavage formation. During cleavage formation, the cell divides without an increase in mass, that is, one large single-celled zygote divides into multiple smaller cells. Planes of cleavage. The cleavage is initiated by the appearance or groove called cleavage furrow. It may divide the egg from different angles or planes. Depending on the portion of the cleavage furrow, the planes of cleavage are named as meridional plane, equatorial plane, vertical plane, and latitudinal plane. In embryo, the lower end is called the vegetal pole, whereas the pole which is opposite to the vegetal pole is called the animal pole. We can also say that the region of the ovum where polar bodies extrude and receives the sperm is known as animal pole. We can see the differences in cell division in animal and vegetal pole. In animal pole, the cell divides rapidly, but in vegetal pole, the cell divides slowly. Based on the animal pole and the vegetal pole, the plane will pass and bisect. Meridional plane. The plane of cleavage lies on the animal-vegetal axis. It bisects both the poles of the egg. The egg is divided into two equal halves, for example, third cleavage furrow of chick embryo. Equatorial plane. Cleavage furrow bisect the egg at right angles to the median axis and halfway between the animal pole and the vegetal pole. Example, fifth cleavage plane of the egg of Ambystoma maculatum. Vertical plane. The cleavage furrow passes from the animal pole to the vegetal pole, but it does not pass through the median axis of the egg. Example, third cleavage furrow of Amia calva. Transverse plane. It is similar to the equatorial plane, but it lies on either side of the equator. It is also called as transverse or horizontal or latitudinal plane. Example, third cleavage plane of frog embryo. Types of cleavage. There are two main types of cleavage based on the axis of division. If cleavage occurs through center line dividing the yolk into two equal halves, it is holoblastic cleavage, and if it occurs along any side producing smaller or unequal blastomere, it is meroblastic cleavage. Both of these kinds occurs in zygote depending upon the amount and distribution of yolk. Based on the amount and distribution of yolk, the eggs are classified into isolecithal egg, which has evenly distributed yolk, mesolecithal egg, which has moderate vegetal yolk disposition, telolecithal egg, which has dense yolk through most of the cells, and centrolecithal egg, in which the yolk is in the center of the egg. In isolecithal egg, four kinds of cleavage are observed, like radial, spiral, bilateral, and rotational cleavage. In mesolecithal egg, displaced radial cleavage is observed. In telolecithal egg, two kinds of cleavage are observed, discoidal and bilateral. In centrolecithal egg, superficial cleavage is observed. Holoblastic cleavage. Radial cleavage. Radial cleavage occurs in isolecithal egg, that is, egg with evenly distributed yolk. Cleavage furrow appears in such a way that each of the blastomeres of the upper tier lies over the lower tier. The cell division takes place so that the cells are aligned directly over each other. The planes of the cleavage are symmetrical to the polar axis. Example, sea urchin, sponges, sea cucumber, and Amphioxus. Spiral cleavage. Spiral cleavage occurs in isolecithal egg, that is, egg with evenly distributed yolk. The planes of cleavage are not symmetrical between the poles. These planes are diagonal to the polar axis. They produce unequal cells around so that the cells are not aligned directly over each other but rather are aligned at an angle. Example, Annelida. Rotational cleavage. Rotational cleavage occurs in isoleucithal egg, that is, eggs with evenly distributed yolk. Zygote divides into two blastomeres. One blastomere divides asymmetrically to give rise to one founder cell for differentiation, and one stem cell with other blastomere divides equatorially to produce normal differentiating cells. Stem cells again divide meridionally to repeat the pattern. This kind of asymmetric cleavage is termed as rotational. Example, mammals and nematodes. Bilateral cleavage. Bilateral cleavage occurs in isoleucithal eggs. First cleavage divides the zygote into two right and left cells, then each right and left side cleave separately on right and left, making mirror image of cells on left and right sides. This is bilateral holoblastic cleavage. Example, tunicates. Displaced radial cleavage. Displaced radial cleavage occurs in mesoleucithal egg, that is eggs with moderate vegetal yolk disposition. It is like radial cleavage, vertically producing same-sized blastomeres. But first, horizontal division is little bit displaced from center towards the animal pole. That is why it is termed as displaced radial cleavage. Example, amphibians. Meroblastic cleavage. Discoidal cleavage. Discoidal cleavage takes place in teloleucithal egg, that is eggs with dense yolk throughout most of the cells. It occurs in fishes, reptiles and birds. Here the cytoplasm is placed at the animal pole as a disc called blastodisc, and only this disc divides. Hence it is said to be discoidal cleavage. The huge mass of yolk does not participate in cleavage. The cleavage division remains confined to the cytoplasmic disc at the animal pole. Example, fish, Danio-Orezias. Superficial cleavage. Superficial cleavage takes place in centroleucithal eggs, that is, eggs with yolk in the center of the egg. Here, the cleavage occurs only in the surface layer of egg and does not extend into the central yolk. In this type, at the beginning of cleavage, the nucleus starts to divide, but it is not followed by the division of cytoplasm. Example, insects, Drosophila. Bilateral cleavage. Bilateral cleavage occurs in teloleucithal eggs, that is, eggs with dense yolk through most of the cell. It is same as in the isoleucithal egg, but occurs only where yolk is low in animal pole. Embryo can be halved into two equal sides as mirror images. Example, reptiles and birds. Cleavage in human zygote. Cleavage in human zygote occurs during its passage through the fallopian tube to the uterus as in other mammals. It is holoblastic. The first cleavage takes place about 30 hours after fertilization. It is meridional, coinciding with the animal vegetal pole axis. It produces two blastomeres, one slightly larger than the other. The two blastomeres remain adhered to each other. The second cleavage occurs within 60 hours after fertilization, and third cleavage takes place about 72 hours after fertilization. Formation of morula. After fourth cleavage, solid ball consisting of 16 to 32 cells are formed, which looks as a little mulberry called morula. Due to holoblastic and unequal cleavage, two types of blastomeres are formed. There is an outer layer of smaller transparent cells called the micromere around an inner mass of larger cells called macromeres. The morula reaches the uterus about 4 to 6 days after fertilization. It is still surrounded by the zona pellucida that prevents it from sticking to the uterine wall. Formation of blastula. The outer layer of the cell form trophoblast or trophoectoderm, which draws the nutritive material secreted by the uterine endometrial glands. The fluids absorbed by the trophoblast collects in a new central cavity called blastocele. As the amount of nutritive fluid increases in blastocele, morula enlarges and takes the form of a cyst and is now called the blastocyst or blastodermic vesicle. The cells of trophoblast do not participate in the formation of embryo. Inner cell mass of macromeres form a knob at one side of trophoblast and is primarily determined to form the body of the developing embryo, so is called the precursor of the embryo. Mammals have a slow rate of division that is between 12 and 24 hours. These cellular divisions are asynchronous. Zygotic transcription starts at the 2, 4 or 8 cell stage. Cleavage is holoblastic and rotational. Humans have holoblastic cleavage with equal division. At the 8th cell stage, the embryo undergoes 3 cleavages where it undergoes some changes. At this stage, the cells begin to tightly adhere in a process known as compaction. Recently, it has been proposed that in placental mammals, the cells become more likely to contribute to one of the first 2 cell types to arise, the inner cell mass or trophoectoderm, depending on their position within the compacted embryo. A single cell can be removed from a pre-compaction 8-cell embryo and used for genetic testing, and the embryo will recover. Most of the blastomeres in this stage become polarized and develop tight junctions with other blastomeres. This process leads to the development of 2 different populations of cells: polar cells on the outside and a polar cell on the inside. The outer cells, called the trophoblast cells, pump sodium in from the outside, which automatically brings water in with it to the basal surface to form a blastocele cavity in a process called cavitation. The trophoblast cells will eventually give rise to the embryonic contribution to the placenta called the chorion. The inner cells are pushed to one side of the cavity because the embryo isn't getting any bigger to form the inner cell mass, and will give rise to the embryo and some extra embryonic membranes. At this stage, the embryo is called a blastocyst.